For the worldwide campaign celebrated on May 16th, we are here to talk about one of the CDG subtypes, SSR4 CDG. CDG stands for Congenital Disorders of Glycosylation. Glycosylation is a process that our body performs by breaking down, transforming, and adding sugar blocks to proteins. All systems of our body are involved, such as the digestive, respiratory, muscular, cardiovascular, and urinary systems. All of our systems need the glycosylation process to function. People with CDG have health problems because their body does not perform this process correctly. CDG are caused by genetic mutations. We can think of CDG as an octopus. Each arm of this octopus is a different disorder that occurs because glycosylation is malfunctioning. But all parts belong to the whole. Each little arm is a different disorder. Every gene is like a book of instructions. Genes determine your eye color, your height, your blood type, and how your cells work. Genes provide theoretically simple instructions so that you can walk, talk, and perform other activities. CDG are caused by a change in these genes, which is like an instruction written down wrong. Because of that, the body cannot read the encoded message correctly, which leads to health problems. SSR4 CDG is part of the CDG octopus, and it is known as a CDG subtype. Our children have SSR4 CDG. The protein encoded by the SSR4 gene can be found in a cell organelle called the endoplasmic reticulum. This organelle is responsible for building glycoproteins. The SSR4 protein is involved in transporting proteins as well as attaching sugar trees, glycans, to proteins and lipids, fats, thus converting them into glycoproteins and glycolipids. Almost 50% of the body's proteins are glycoproteins. They are needed in almost every organ cell, so almost all CDG patients have multi-organ problems. How does this happen in SSR4 CDG? Unlike most types of CDG, most SSR4 cases reported so far are due to a de novo mutation. That is, the mutation occurs spontaneously in the patient. In this case, the mutation is not inherited from the parents and occurs during embryonic development. As the SSR4 gene is located on the X chromosome, SSR4 CDG is an X-linked recessive disorder, and all males have only one copy of the defective gene. Female carriers of the defective gene can also show mild symptoms associated with SSR4 CDG. 15 males have been reported with a confirmed diagnosis of SSR4 CDG as of May 2023. There are likely to be unreported others. Additionally, we estimate that many remain undiagnosed due to the lack of awareness of SSR4 CDG within the medical and scientific communities. Lack of information about the complexity of this disorder results in difficulty accessing the diagnosis. Clinicians submitting genetic profiles of their patients to the public archives help us identify families with SSR4 CDG family members. According to ClinVar, a public archive with free access to reports on human variations of congenital genetic mutations, 5 interpretations of new SSR4 variants have been submitted just in the last 2 years. We see that there are 2 boys identified in China, 1 in California, 1 in Texas, and 1 at a medical center in Israel. The dates represent when the variant was reported or updated. SSR4 CDG is a growing community with new patients being diagnosed each year. Hopefully, this fact will encourage research scientists and clinicians to study SSR4 CDG. Unfortunately, the biological mechanisms of this CDG are not yet well known. The protein encoded by SSR4 is part of a complex called the translocon-associated protein, TRAP. The translocon system is present in every cell of the human body, on the membrane of an organelle called the endoplasmic reticulum, ER. The translocon allows the proteins being built from amino acids to enter the ER lumen and undergo glycosylation, as you see in the figure on the left. This complex likely cooperates with two other complexes, the SEC61 channel and the oligosaccharide transferase, or OST, complex. The OST is an enzyme responsible for glycosylation. Therefore, defects in the SSR4 protein may result in the OST being unable to glycosylate proteins, resulting in lack of glycoproteins, or hypoglycosylation, in the SSR4 CDG syndrome. It is also speculated by scientists that TRAP may have a role in the identification of misfolded, damaged proteins, which need to be degraded to keep the cell safe. TRAP preferentially binds misfolded proteins, according to Nagasawa et al. 2007. The figure on the right is a microscopic close-up of the TRAP complex on the endoplasmic reticulum membrane. The signal sequence receptor 4, or delta subunit, is one of 4 subunits of the TRAP protein complex. It is circled in red. The main clinical symptoms of SSR4 CDG are developmental delay, intellectual disability, hypotonia, seizures, facial dysmorphism, skeletal anomalies, failure to thrive, strabismus, problems with feeding, gastroesophageal reflux, and other abnormalities. Currently, there is no specific treatment, but there are some clinical management strategies. These therapies can prevent complications associated with the disorder and can stimulate the development of patients. A multidisciplinary team of doctors and other health professionals is needed. Children with CDG SSR4 benefit from physical, occupational, and speech therapy. Currently, there is no effective drug treatment. The scarcity of CDG SSR4 cases is a challenge for research. We believe in science, and as studies continue to evolve, we dream that one day a treatment will help their bodies make what they cannot synthesize naturally. Life is not what it should be. It is what it is. It is how we deal with it that makes the difference.